Hey everyone, I'm pleased to present our work titled Finally Bounding Box is All You Need, Learning to Segment Cells into Microscopic Images for Box Annotation. My name is Nabil Khalid and I'm presenting this on behalf of my co-authors. Thank you very much for attending. So before getting started, let's discuss the importance of cell segmentation. Cell segmentation is crucial in biological research and medical applications. It helps us understand biological processes and disease at cellular level. Accurate cell segmentation aids in disease diagnosis by identifying abnormal cellular phenotypes, and it is essential for drug discovery and screening, helping to identify cellular morphology and spatial organization. So there are certain challenges associated with annotating the microscopic data. Traditional method need extensive annotated data, and manual annotation is labor intensive and very costly. For instance, annotating cells in a live cell dataset takes about 46 seconds per cell, highlighting the inefficiency of traditional methods. So in order to overcome that issue, we propose time-saving solution which requires only the bounding box annotation, significantly reducing the annotation time. This approach is approximately 7 times faster than the traditional manual segmentation methods. By using only bounding boxes, we streamline the workflow and reduce overhead, making the process more efficient. Now we will discuss different kinds of training supervision for cell segmentation. The first one is mask-based, which requires full mask for training. It was basically it's very time-consuming and it requires expert knowledge incorporation as well. And it requires 46 seconds to draw segmentation mask by itself. The second one is point and box based from the paper point to mask, which requires a bounding box and some point annotations inside the bounding box. Those point annotation can either be on the cell or outside the cell. If that the point is on the cell, it's shown as blue dot, and if it's outside the cell, it's shown as red dot. So it's basically a two-step process in which annotators first draw the bounding boxes and then the points are generated automatically inside those bounding boxes. And then again, the annotators have to decide whether that point is on the cell or outside the cell. But this process can get very confusing because of the fact that if the cells are in close proximity of each other, like you can see here, it can get very confusing for the annotators and because the foreground for one cell can be the background of another cell. So it can cause a lot of overhead on top of the time which is mentioned here. So to overcome that issue, this paper proposed a pipeline called Cellboxify, which requires only the bounding box based annotation, which streamlines the annotation process by a great margin, and it requires only 7 seconds per cell to annotate the data. And it significantly reduces the expert knowledge which is required and significantly cut down the annotation time. Now we'll discuss the different annotation time required for different trainings supervisions. For full mass requires 46 seconds for per cell, where there is point to mask requires 7 seconds for burning box, and depending on the number of points, the time is also added up. So, for example, for 6 points and a burning box, it requires 14.2 seconds, and for cell boxify, which requires only the bounding box, it takes only 7 seconds, which significantly reduce the time. So now let's dive into the details of our cell boxify pipeline, which is designed to efficiently segment cells in microscopic images using only bounding box annotations. And the cell boxify pipeline consists of three main components: the backbone network, the regional proposal network, and the prediction head. Each component plays a crucial role in achieving accurate cell segmentation. The backbone network is responsible for extracting feature maps from the input image at different scales, and then those features are passed on to the region proposal network, which extracts the regions which contain objects and passes it down onto the prediction head. The final component is the prediction head, which refines the bounding box to proposal and generate accurate segmentation marks. The prediction head in our pipeline utilizes mask arsenal architecture, extending the capabilities of faster RCN and making a branch of predicting segmentation masks. It integrates the point-based unary loss, and distance-aware pairwise loss to optimize the segmentation predictions. The point-based unary loss ensures that the vertices of the predicted polygon falls within the ground truth bounding box, minimizing discrepancies. And the distance-aware pairwise loss enhances the accuracy of object boundaries by considering local and global variations, ensuring smooth and precise segmentation. To handle memory constraints, a clipping strategy is used, resizing the predicted polygons to fixed size, making the process more efficient. So to summarize, the integration of these components in the cell boxify pipeline allows us to achieve high-quality cell segmentation using only bounding box annotations. And this methodology significantly reduces the time and expertise required for annotation while maintaining the strong segmentation performance. And by leveraging advanced deep learning techniques, we streamline the segmentation process, making it scalable and practical for large-scale biomedical image analysis. So for this study, we utilized the live cell dataset, which is the largest cell biology dataset with 1.6 million cells across 5,239 images. It features an average cell density of 313 cells per image, which surpass other comparable datasets. The dataset includes distinct cell cultures reflecting real-world cellular environments. And we utilize only the bounding box annotation for this dataset. And we evaluated our method using standard cuckoo evaluation protocol, and we are reporting mean average precision, which is the precision average across all unique recall levels. Now this slide presents the quantitative performance comparison between full mass, point to mass, and cell boxify on the live cell test set. The full mass method achieved segmentation AP of 43.9, and the point to mass achieved 43.5, and our proposed approach cell boxify achieved 36.6. Despite only using bounding box annotations, cell boxify reached 3.40% of the performance of the fully supervised method, demonstrating its effectiveness. And here we compare the segmentation performance across different cell cultures using full mass, point to mass, and cell boxify. And boxify performed best with SKBR3 and BV2 cell cultures, closely matching the performance of point to mask and full mask. However, it showed lower performance with SHSY cell culture due to its complex neuronal morphology, highlighting areas for future improvement. And here are some of the qualitative comparison. Here the solid yellow lines represent the ground truth, and the dotted red lines represent the predictions made by the model. And you can see cell boxify performs at par with the full mask and point to mask. Similar results can be seen here. So to conclude, our cell boxify method reduced annotation time by 85% while achieving 83.4% of the fully supervised performance. We streamlined the annotation process, making it practical for large-scale tasks and challenging scenarios. And the method maintains strong performance across various cell type and imaging conditions, demonstrating its versatility. Thank you very much for listening.